Cool. Welcome, everyone. My name's Nabila, and I'm a stream owner here at ACE. ACE is a community of machine learning practitioners and researchers who have gathered around topics in AI research, engineering, and products. We host free live sessions like this about 3 to 5 times a week. We also produce premium content in various subject areas. We currently have 14 different streams that are focused on different ML topics. This particular stream is GNNs for healthcare. We hope you enjoy our talk. Today we're speaking with Adnan from the University of Waterloo. Adnan is a graduate student at the Chemia Lab at the University of Waterloo/Vector Institute. He was recently at the National University of Singapore Hospital as a research intern. He received his bachelor's degree in electronics and electrical engineering at IIT, and his current major research interests include statistical machine learning, probabilistic deep learning, and their applications in vision. Today, Adnan is presenting representation learning of histopathology images using graph neural networks, which he is also presenting at CVPR this year. So over to you, Adnan. Welcome.
Thanks, Nabila. Hello, everyone. Today, I'm going to present my recent work on representation learning for pathology images. So we submitted this paper to CVPR 2020. So representation learning is a very big field, and especially for histology images, it's very challenging to do so because of its large size and other complexity issues. So in this work, we propose to develop a very efficient way to extract features for histopathology images, which can be used for classification, image search, or other purposes. So let me just a second. So let me give a brief introduction to pathology and how the biopsies are captured. So recently, the way biopsies are captured or scanned have changed drastically with the introduction of digital pathology. Now, pathologists are using digital scanners to scan the whole site images and store it on a computer, which can be shared with different pathologists located in different hospitals in different city or maybe in different countries. This is very different from the conventional approach. So earlier, pathologists used to manually see these slides using microscope, zoom in different location, and analyze this whole slide manually. So, but with the scanner, we can basically scan the whole slide and visualize the image on the monitor. So the problem with this technique is that each slide is about 2 GB in size. If we compare to now conventional data set for vision, which is like a few GBs, not more than few GBs for the entire data set. And now we have a single WSI whole slide image, which is itself a 2 GB or 3 GB image. So in terms of data size, a historical data set can be as big as 1 TB or 2 TB, which is a very enormous size. And so in order to extract feature from such a large data set is a very challenging task. So digital pathology has created a very good avenue where we have large archives of scans available. And there's a massive information stored in such archive in different hospitals. However, there has not been any efficient way yet to extract this information, which can be used for diagnostic or research purpose. And the main reason for this is the size of each WSI, which contains billions of pixels. And so therefore, conventional CNNs cannot be directly used for such big image. So the recent advancement in deep learning has opened very promising field in data pathology. Now, pathologists are very excited to use AI or deep learning and see whether they can build some system where WSI can be automatically classified into cancer and non-cancer images. But since the size is very large, so most of the popular approach just downsample the WSI into a very small image and then train the simple network. But if we do so, the main problem is that we lose a large amount of information in downsampling. So one way to tackle this problem could be MIL or multi-instance learning. So multi-instance learning is exactly where this problem for histopathology fits. So each WSI can be thought of as containing multiple patches. So we can extract, suppose, 500 patches from a WSI and then train a multi-instance learning algorithm. So in multi-instance learning algorithm, we have a label for a group of patches, a group of instances, instead of a single instance in conventional machine learning approaches. So this figure will explain the difference between conventional machine learning and multi-instance learning. So in multi-instance learning, we have a group of objects or instances, and we have label for the entire group, not for the individual instances. Where if we look into conventional machine learning, we have label for each instances, and we train a network. So this is very relevant for medical imaging. So if we think as a WSI as a group of patches, and each group can contain about 200 or 300 patches, and we have a label for the entire group of patches, that is WSI, whether it's a cancer or non-cancer image. So multiple instance learning is a very promising approach for learning histopathology representation. There have been many works on multi-instance learning for pathology images, but most of the approaches assume that the data or the patches are independent of each other, which is not very true, because there's definitely a spatial relation between the patches. And even if the doctors thought when they analyze the slide manually, they try to develop a connection between different patches and then come up with a decision. So where currently the approaches that are being used for MIL assume IID nature, that's independent and the patches are independent. So we improvise this algorithm with the use of graph neural networks. So we propose that each WSI can be transformed into a graph, where each node in the graph is a patch from WSI. So suppose I have this WSI, I extract 100 patches from it, then each patch is a node in the graph. And our goal is to convert this WSI into graph structure. And then this graph can be fed into a graph neural network to get the representation. But the challenge is that we don't know how the relation between patches is. So we propose a two-stage method. So the first stage is that we convert the WSI into a graph, and second stage basically use a graph neural network to get the representation for the WSI. So in the first stage, we extract multiple patches using color-based extraction methods. So a single WSI can easily yield up to 200 to 500 patches from a single WSI. So we have nodes which where each node is a patch, and then we extract feature for each node using some pre-trained network. In our case, we use DenseNet, but it can be any other trained network. Once we have the feature for each patch or each node, we pass this feature to a density matrix learning layer. So a density matrix is basically describe how patches are connected to each other. So for a graph, a graph can be fully described using two variables. One is node list and one is adjacency matrix. So node list is just basically the patches that we have in the graph, and adjacency matrix describe how these patches are connected, like if patch 1 is connected to patch 2 or not, and so on. So we introduce this adjacency matrix learning layer in a network to learn the dependencies between different patches and transform the WSI into a graph representation. So we use something called global context pooling. So the first layer, the first part of this layer usually condenses the whole WSI into a single vector. So we aggregate all the patches into a single vector using a very basic aggregation method. This is being done so that we know the context of the WSI, whether like what's WSI is all about. And then we just concatenate this context vector with the initial feature vector. So the vector we obtain here is just concatenate version of these two objects. And then we do cross multiplication to get the adjacency matrix. So at this stage, we have a graph representation of WSI. We have a node list V and a density matrix A. Now, task is very simple. Now we can use.
Sorry, Adnan, can I interrupt you with a question?
Sure.
Cool. So this, to actually do one round of this, you need to have an adjacency matrix, right? And you're saying that we don't actually have this information before you start learning?
Yes, that's right.
Could you not use the fact that patch 1 is like top left corner and patch 2 is a little further away? Could you use those distances from the actual spatial image to start the adjacency matrix?
We could, but that's not the best approach, in my opinion. First of all, WSI is not very oriented image. Like this left corner can be left for you, and if someone else can decide, it could be in bottom right. So spatial location is relation is there. That's true, but we don't know like how it's the best way or not. So we randomly initialize agency matrix, and we train in end-to-end fashion. So the whole network is trained in one go. So in order to get the best representation, agency matrix has to learn the actual relation between them. And it could end up learning spatial relation if that's the case.
Okay, so in the perfect scenario, you would want it to learn importance to neighboring patches and not actual spatial structure?
Yes, that's right.
Okay, cool.
Because I have cancer patch here and a cancer patch at the opposite corner. Definitely they are more connected than the neighboring patches. And that's why we don't want to use spatial information as a prior for the agency matrix.
Okay.
So at this point, we have a graph representation for WSI. So now we can use any graph neural network to get the representation of this graph structure. So most conventionally, graph neural network are used for social network graphs or recommendation system. But we propose that graph neural network can also be used for the WSI. And that's very relevant for WSIs because we can construct relation between different patches. Right now, most of the approaches label the small patch with the label of the WSI and then train CNN, which is not the very optimized way to go. So we try different types of graph neural network to see which works best for WSI. So we experimented with two kinds of GCNN, spectral GSN and spatial GCNN. And we performed a very brief ablation study like spatial works better or spectral works better. And we found that spectral GCNN works better and achieves a higher accuracy for the classification task, where spatial GCNN doesn't perform that good. So definitely, spatial information is not it's not very optimized for getting the right agency matrix. So once we have the graph structure, we can just use any GCNN. In our case, we found that spectral works better. So we have an input graph of WSI, which was learned in the previous slide, in the previous network. Now we have multiple layers of graph convolutions. So the graph is transformed into different feature space, and which finally pooled using graph pooling layer. So we tried different kinds of pooling layer here, and interestingly, we use global attention pooling, which can give attention score for each patch. So after we pool the graph into a single vector, we can then easily use it for classification using any dense network easily. So the most important innovation in this entire thing is the agency learning layer, because we don't have any prior knowledge about the connection. So the main task is to learn the agency matrix first, and then we can use any GCNN. So we introduce global context vector, and we do so because there might be a case where two patches are present in multiple WSI, but their relation depends on the can be different for each WSI. So that's why we want to first calculate the global context vector, which basically tells about the entire WSI briefly. So and then we start building the each connection. So and in pooling layer, we try to use different kinds of pooling methods. So popularly, people use global attention pooling or mean pooling, max pooling, and sum pooling. So we tried different approaches and performed the ablation study and compared each pooling layer with other. So the benefit of using global attention pooling is very interesting. So if we use global attention pooling, we can basically get attention score for each patch. So this is very useful for pathologists. Especially right now, pathologists are very reluctant to use deep networks because they are very kind of black box. But this approach can tell the pathologist like, okay, this patch is more important because it has high attention score. So this is a more transparent way to uh, go to do classification, and inference is very easy for using this kind of network. So in our experiment, we use lung cancer subtype classification task. So our aim was to classify lung cancer into two subtypes, LU80 and LUSC, which are the most difficult types of cancer to classify. Even pathologists have hard time classifying these two cancer. So this is how each patch looks like. So we have about 500 patches for each WSI, which looks like this. And so we start building a graph structure for using these patches, using adjacency matrix learning layer. And once we have the graph structure, we can just feed it to graph neural networks to get the representation. So we found out that using our approach, we got state-of-the-art accuracy. So we didn't train any network from scratch. We just used pre-trained dense network, and still we could achieve AUC value of 0.89, which is a state-of-the-art right now. So for experiment, we used the largest dataset available on public domain, the TCGA. So we almost extracted about 2,000 patches from dataset, 2,000 slides from dataset, and trained the network on it. So as you can see, like the previous approaches have didn't perform that well compared to our method.
Adnan, just a few questions on the slide.
Okay.
The baseline methods you're comparing to, are any of those graph methods?
No.
No? Are they traditional computer vision? Can you say more about what those baselines are? Like, they're state of the art right now?
These baselines?
Yeah.
One of them is use multi-instance learning, but not graph networks.
Okay.
And other you just use train network from scratch, but none of them is graph neural networks.
Oh,
I think we are the only paper that use graph neural networks for the pathology images.
Very cool. Can you say more about the labels that you used? So if you take a WSI and you split it up into patches, you said that you don't just use the overall WSI label as a patch label, right?
Yes, exactly. So in our experiment, we use binary classification. That was one class was LUAD and other was LUSC. So we were not classifying into cancer and non-cancer images. We were classifying into two subtype of cancer, whether it's LUAD or LUSC. So we extracted multiple patches from a single WSI. And for the entire group of these patches, we had a single label. So that's what MIL needs. MIL, multi-instance learning, needs a label for the entire group of patches instead of single instances.
Okay, so in a bag, you would have some negatives as well.
Yes, like some of the patch could be just fat tissue or something like this, which is not really useful.
Okay, got it. Thank you.
So the main benefit of using global attention pooling is the inference. So after we train the network, we can easily do inference by getting the attention score from the network. Suppose if I give a slide as an input to the network, I can get attentions value for each patch individually. And this information can be very useful for the pathologist, as they can see which patch is the most important one according to the network and verify is it true or not. So we show a very small example. So we do inference for 2 WSI. So these are the patches which got the high attention values, and these were the patches which got very low attention values. And very interestingly, that we saw that patches with high attention value consist of have high nuclei rate than the with lower attention, and this is very true. Like even the pathologist is focused on patches where they have more nuclei, and this property was learned by the network on its own. So this is very cool application of global attention pooling, and definitely it can help pathologists in getting a more explainable AI solution for histopathology. So this is a very brief ablation study we performed. We experimented with spectral GCNN and spatial GCNN. So the spectral one we use a ChebNet, and the spatial one was stage convolution. So as you can see, that spectral outperformed the spatial one with a huge margin. And we tried different pooling layers, and we tried different combination of spectral and different pooling layers. So we found that spectral GCN with mean pooling layer works best, but it's not very different than the attention one. So the benefit of using attention is the inference one, and the accuracy is not that low compared to mean. It's a very small difference. So the basic conclusion of the whole paper is that we propose a very novel architecture for learning representation for the histopathology images, and it's somewhat explainable. We can infer what patches are important in the entire WSI, and this could give more information to pathologists to make a correct decision. We can even visualize the agency matrix and see how patches are connected to each other, and and we can get a single vector for the entire WSI, which can be further used for other tasks like image search or any other learning task. So we also did a very closely related work called learning permutation event representation. In this paper, we use something very similar to this GCNN, but we use memory networks to learn the dependencies of different patches. So this could be interesting. Who's interesting too?
Adnan, which memory networks are these? Is this the new iClear paper?
Oh no, it's the conventional memory networks.
Okay, I'll have to look it up. Not familiar with it.
The paper, so maybe those who are interested in this paper can find this also useful.
Definitely.
So that's all about the paper. I hope we can connect, and if you have any questions, let me know. If you have questions later, ping me on, email me, or add me on LinkedIn. Anyway.
Awesome. Thank you so much for your slides. We have just a few questions. I guess if you could go back to your main slide about the pipeline, where you go through the node representation, then the adjacency learning. I think it's like slide 10 or something. No, sorry, it's a little before that. I don't know the number. Yeah, that guy. Cool. So there are just a few questions that we have over here. Just give me 1 sec to pull them up. So I guess one question that I have actually is, do you do any initialization on the adjacency layer? Because you are treating it as a learnable parameter, right? So where do you start?
It's a random initialization, like we do in our deep network. We don't initialize for spatial information or as such. It's completely random.
Okay. The reason I ask is we've had a similar talk on this stream, where the adjacency matrix was initialized because the training wasn't stable. So between every layer, the adjacency values jump quite a bit. I'm wondering if you've had that, if you see that happening here. Is there any way to constrain the learning of the adjacency matrix?
No, we don't put any constraint on the agency learning layer. I think the cost function value is decreasing. That means the agency matrix learning layer can converge to a local minima, right? So
Right.
that's what we hope. And
Okay.
I've seen some papers that use a normal L2 distance as a way as matrix. But if we use L2 distance, that's very I think simple approach. Like we just use color information to get the relation between patches, right?
Hmm.
So that's why we didn't initialize with any kind of L2 distance or spatial information and so on. We just randomly initialize it and then use cost function to convert the matrix to a local optima.
I see. So I guess at the end of your training, how do you know if it's an appropriate adjacency matrix? Is there a way to because in a typical graph setup, it's correct if you can see connections, right? So if I'm currently connected to the neighbor behind me, then that's probably good structural information that I learned through this process of supervision. I'm wondering, have you guys done any of those tests to figure out if it's correct? Like, how do you know those values are accurate?
Yes. So one thing is that if the adjacency matrix was not accurate, we would not be able to achieve good accuracy, right?
Mm-hmm.
We have visualized the adjacency matrix, and we are currently showing to different doctors to see whether it makes sense or not. But we hope that there is some information, useful information in the adjacency matrix, because otherwise we were not able to classify any WSI with any random graph structure.
Right.
But
Okay.
being engineers, we don't know about medical science, right? So we are showing to different doctors right now to see whether it makes sense or not.
Okay. And what would make sense most like to the doctors in terms of this matrix? Like that I should be connected, if I'm the center node, I should be connected to all my neighboring nodes, or I should be connected to only like the top left ones, or is there some sort of domain knowledge you have on this area here?
Yeah, according to my understanding, suppose I have two C patches that contains cancer, right? So those patches must be connected to each other. And suppose I have a neighboring tissue here, which is a fat tissue. It should not have any relation to other patches, right? So that's what we really want to explore with pathologists, like the
Okay.
way the pathologist makes graph structure when he examines the slide manually, and the way the network does. Is there any similarity? Does it make sense or not? So we're currently working with few pathologists.
Okay. Interesting. Could you say more about why the motivation for your global context pooling?
Sure. So the idea is that suppose I have two patches, patch one, patch two, and let's assume I have a second slide, WSI, in which patch one, patch two are also present there. But the relation between patch one and patch two depends on the entire WSI. So we need to condition the relation between each patch on the information present in the whole of WSI, right? So that's why we extract global context vector, which basically aggregates all the information present in WSI and condition the vectors on it. So
Okay.
the matrix I get here could be different for different patches depending on different WSI.
Okay. So you want to give it a better chance of learning structure by giving it the initial, the whole image's DenseNet features.
Yes.
Okay, makes sense. I guess questions about your results. Do you have any intuition why the spectral methods are super outperforming GraphSage?
I think spectral methods tries to build a spatial connection, right? So, mm, and I think that could be one reason. Like spatial information, it doesn't really it's not very useful for histopathology. Maybe that could be the reason, but I'm not sure about it.
Yeah, I know it's always hard, right, to figure out what
Yeah.
they learned, but at the end of the day,
It's
it's
a
just very
it's an
surprising.
empirical study, right? So we can just do experiment and see which works better.
Yeah, definitely. Cool. Just curious, what types of aggregators did you use in SageConv?
Sorry.
What types of aggregators did you use in the GraphSage
Actually,
for the neighborhoods?
for the neighborhoods.
Yeah, like you do average aggregation or LSTM or max?
Oh, I think we use LSTM. I don't know exactly. So we use a library called PyTorch Geometric.
Right.
So we use that library. I think by default it's LSTM.
Oh, okay, interesting. Just curious, because the original paper said max was their best, so I just wanted to know if that's a hypothesis everyone is facing as well or not. But I'm just curious.
I didn't explore that thing, to be honest. Like,
It's fine.
we use a default setting in the stage convolution.
Cool.
As the main goal was to compare spectral and spatial, not stage with different configuration of stage convolution.
Yeah, SageConv is typically a good baseline, right? So one of the first spatial ones to be released. Cool. So what are your next steps here? Would you think you guys will roll this out into clinicians, like a clinical testing sort of phase?
Yeah, as I said, right now we are trying to verify our result with pathologists. Like, there's a lot of things to do we can do with agency metrics. So we are showing our result to different pathologists, and based on that, if they think that agency metrics make sense, then we can try to implement in different hospitals. And our lab is also part of ORF fund, which like at the end of 5 years, we want to implement a software in different hospitals for histopathology. So it could be part of that software eventually.
Wow.
Pathologists say the agency metrics make sense.
Interesting. I'm just going to do a last round for questions. If anybody has some, please feel free to populate in the YouTube chat. Maybe we'll give it a few seconds. I think that's all the questions that our audience has for today. Thanks so much, Adnan, for being with us. Your work is really interesting and it has very strong implications for the healthcare space. So it's really interesting to see it come up. For the audience, if you're more interested in this talk, you can visit our website at ai.science and you'll have more access to the slides that Adnan has created for us. Also, make sure to subscribe to ML Papers Explained to get notified of all our live sessions and other free content. Cool. Thanks so much for joining us.
Thanks, Nabila. Bye.